Welcome to our review on binary fission. When we're talking about binary fission, what we're referring to is the process by which prokaryotic cells are going to replicate. And what they do is they copy their genetic material and then split to make two identical daughter cells. When we're looking at the speed at which this can happen, it's actually really quick. So with some bacteria, this will happen every 20 minutes. If we just go back to understanding this phrase binary fission, then you can actually understand these scientific words, which are a little bit strange, by breaking them down into easier to understand chunks. So bi means two, so binary is all to do with two, and then fission is all to do with splitting. So remember, fission with the two S's in the middle is all to do with splits, with the S's at the front and the end. What we need to do now then is understand what's happening through this process of binary fission. And there are 4 stages that you need to remember what's happening in. So stage 1 is where the circular DNA and the plasmids replicate. And you can see that in the diagram there in stage 1 and stage 2. Step 2 is where the cell gets bigger and the circular DNA moves to the poles, which are the ends of the cell. And you can see that in the 3rd diagram. Stage 3 is where the cytoplasm begins to divide and the new cell walls begin forming, which you can see in 4 in the diagram there. And then our final stage, stage 4, is where the cytoplasm fully divides and our 2 daughter cells are produced. So what we find here is that we've got the same circular DNA in each daughter cell, but the number of plasmids can actually vary in each of those cells. So you might find 1 plasmid in one of the daughter cells and 3 in the other, for example. When we think about the population of our bacteria, what we can actually see is that they can divide really quickly if the conditions are right. However, if those conditions for any reason become unfavourable, then the bacterial cells will start to die. What we can do is show these population changes on a graph like the one on the right there. Good news, GCSE, you don't have to know all those phrases. This is just a very clear graph that I found to show you, but you don't have to know everything. The key bit to know about is this bit called the exponential phase on the left there. So this is where the bacteria are doubling in number with each division, which remember could be every 20 minutes. So you can see how the population increases really rapidly then. The other bit you should be aware of is on the far right, that death phase, which is where the conditions have become unfavourable, so the number of cells is then decreasing. One of the things they could ask you to do is to actually calculate the population of bacteria present after a certain period of time. So what they'll actually give you in the question is all the information you need to do this. You'll have your starting number of bacteria and you'll have the mean division time. If we have a look at how to do this with an example then. A starting population of 2 bacterial cells has a mean division time of 20 minutes. How many cells will have been produced after 3 hours? So there are 3 stages to work this out. Stage 1 is where we're going to convert our times to the same units. So we've got minutes for our mean division time, hours for our total time, so we convert those hours to minutes. So 3 hours is 180 minutes. You just do number of hours times 60. Stage 2 is where we're going to work out the number of divisions that have occurred in that total time. And what we do is the total time divided by the mean division time to get that. So 180 divided by 20 gives us 9 divisions have occurred. Our 3rd and final stage then is that we need to multiply the original number of cells by 2 for the number of divisions we've seen. So you could do this as 2 times 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 2, which gives you 1024, which you can just sit and do on your calculator. Or you could do 2 times 2 to the power 9. So all you need to do is 2 to the power of however many divisions there were, and then multiply your original number by that. So that's the easier way to do it in an exam to save potential errors in counting. If you want to just literally do it as doubling it each and every time, you can, but think about which way is more appropriate for you as an individual. Hopefully at the end of this video you can describe the process of binary fission. You can explain how that impacts the change in the number of bacteria in a population, and you can also calculate how many bacteria there would be in a population after a certain period of time.